Mention that COVID-19 has had since its debut, one might believe that everything that the media has conveyed on screen is what we know of the SARS-CoV-2. However, it is important to highlight that COVID-19 comes from a family known as Coronavirdae, which is rich with knowledge beyond what we hear and see, a truly subanatomical gem worth diving into. We'll go over some areas of the infectious diseases field in future videos, but just as an introduction to what a virus is, it is important to acknowledge that a virus is a non-living submicroscopic infectious agent which replicates inside living cells for survival. The major difference between virus and virion is that virus is the nucleoprotein particle, while the virion is the active infectious form of the virus. In other words, when outside of an infected cell or in the process of infecting a cell, viruses exist as independent particles known as virions. The Coronaviride family are large, enveloped, single-stranded RNA viruses, meaning that their nucleic acid material is made out of ribonucleic acid, which is long for RNA. Compared to DNA, which is deoxyribonucleic acid, RNA stands out in three ways. It is usually a single-stranded molecule in its biological form. RNA contains ribose backbone compared to deoxyribose, and the complementary base to adenine in DNA is thymine. However, in RNA, it is unmethylated form of thymine, which is uracil. I'll go over some basics in genetics in some other videos in the future. Coronaviride are the largest known RNA viruses, with genomes ranging from 25 to 32 kilobase. The family contains two subfamilies, the Coronavirinae and the Torovirinae. It is important to note that toroviruses are currently being classified under the family Tobanniviridae. However, in the past, due to similar structure, replication behavior, and genetic sequencing, the toroviruses have been classified or lumped into the Coronaviridae. And thus, for the sake of this video, we will talk about the toroviruses under the umbrella of the Coronaviridae. Coronavirus virions are roughly spherical and known for the large spike S glycoprotein which extends from the virus envelope. All coronaviruses encode four structural proteins, three membrane-associated proteins in the S, M, and E, and a single nucleocapsid protein which is the N. However, some of the genus Beta-coronavirus have an additional membrane protein with extra functions known as hemagglutinating and esterase activities, thus the HE proteins. The capsid protein N is bound to RNA genome and makes up the nucleocapsid. It is effective in replication of the virus as it packages the genome into the virions. The spike protein S is important for binding to host cell receptors for viral entry. It binds to a receptor known as ACE2. It has two components in S1, which regulates the viral host interaction, and S2, which mediates fusion and viral entry. The envelope protein E and membrane protein M are important in the formation of the viral envelope, with the latter being involved also in the general coronavirus assembly and integration. Within this envelope is a flexible or doughnut-shaped nucleocapsid depending on the subfamily that consists of genomic RNA. The tubular nucleocapsid is bent into an open torus, thus the name a torovirus. Toroviruses have a glycoprotein with HE activities. HE mediates binding to sialic acid residues and cleaving them. Sialic acids are acid sugars with a carbon backbone, serving as parts of the likes of glycoproteins and glycolipids. Some scholars consider that HE allow toroviruses to penetrate into the mucous layers of the respiratory and digestive tracts. Members of the subfamily Coronavirinae are widespread among different living beings with mainly mammals. They induce mild respiratory or enteric symptoms. Over 60 coronaviruses have been isolated from bats, and most of these are in the genus Betacoronavirus. Bats act as large and mobile bioreservoirs, some bat species having unique viruses which can go back to a probable history of coevolution. Toroviruses have been isolated from humans, cattle, horses, swine, goat, and sheep as they cause symptoms of gastroenteritis. This subfamily is difficult to grow in cultured cells and thus has not been adequately studied. This ends part 1 of this video. I highly suggest going over this article if you are interested in this topic. Thank you everyone for watching and I hope you learned something new today. If you enjoyed the video, make sure to like and subscribe to my channel. Take care and have a good day.